This tiny teach is about DNA replication in bacteria. Bacteria must replicate or copy their DNA before binary fission. Basically, bacteria trace both strands of their DNA to make a copy. That's the easiest and most accurate way to copy DNA. This is known as semi-conservative replication. Both of the original DNA strands serve as templates to produce a new DNA strand. In the end, we will have two copies of the DNA. Each copy is half original DNA and half new DNA. In prokaryotes, DNA replication occurs bidirectionally from a single point, the origin of replication or ORI site. Helicase unwinds and unzips the DNA at the ORI site. ATP is required for this step. DNA replication then proceeds bidirectionally around the circular bacterial chromosome. Single-stranded binding proteins hold the two DNA strands apart and prevent the DNA from zipping back up. DNA polymerase adds complementary nucleotides at about 1,000 per second in the 5' to 3' direction. DNA polymerase also proofreads as it builds new DNA strands. Since the two DNA strands run anti-parallel, one runs 5' to 3' and one runs 3' to 5', the strands will have to be copied slightly differently. One of the DNA strands is replicated continuously. It's known as the leading strand. It can be done continuously because DNA polymerase can add nucleotides in the 5' to 3' direction continuously. The other DNA strand is replicated discontinuously in fragments. It is known as the lagging strand. The lagging strand is replicated in fragments known as Okazaki fragments that are about 1,000 nucleotides long. The strand has to be done this way because DNA polymerase can only add nucleotides in the 5' to 3' direction. The Okazaki fragments are knitted together by DNA ligase.